Mycobacteria are slim, gram-variable rods that do not stain well with the gram method because of their high lipid content in the cell wall. Instead, we use acid-fast stains. The Ziehl-Neelsen stain is considered the hot method, while the Kinyoun stain is the cold method. Another option is the auramine-rhodamine fluorescent stain. All of these rely on the presence of mycolic acid, a waxy lipid in the cell wall. Importantly, the presence of any number of organisms on a smear is considered significant. When growing these organisms, we use specialized media. Lowenstein Jensen medium contains 60% egg in its nutrient base and includes malachite green. It is solidified into slants after heating to coagulate the proteins. Tween 80, which is oleic acid, helps disperse colonies in liquid media. Carbon dioxide is particularly important in the first 24 hours of incubation. Most mycobacteria grow at 36 degrees Celsius, though some require 30 degrees. Growth is very slow, often taking 3 to 6 weeks, though automated liquid culture systems allow faster growth and susceptibility testing. Identification can be confirmed with nucleic acid probes. For specimen collection, the preferred sample is sputum, ideally collected as the first morning sample on 3 consecutive days. Bronchial washings, gastric washings, urine, and tissue are also acceptable. Samples must be collected aseptically into sterile, tightly capped containers. They may be refrigerated overnight, but gastric and urine samples should be neutralized if held. Specimens require decontamination to eliminate competing bacteria. The most common method uses N-acetyl-L-cysteine, or NaLC, combined with 2% sodium hydroxide. Trisodium phosphate and benzalkonium chloride, also known as zephyrin, may be used when Pseudomonas is present. Dithiothrytol with 2% sodium hydroxide is a comparable method. Strong alkali, such as 2% sodium hydroxide alone, acts as both a mucolytic and antibacterial, but exposure must be limited to 15 minutes as prolonged contact can kill mycobacteria. After treatment, the specimen is centrifuged for 20 minutes at 3,000 revolutions per minute, and the sediment is used to prepare smears and inoculate media. Branching gram-positive rods include Actinomyces and Nocardia species. Actinomyces israelii grows best under anaerobic conditions. For biochemical reactions like casein, tyrosine, and xanthine utilization, these tests are not applicable. This organism does not demonstrate acid-fast characteristics. Nocardia asteroids, on the other hand, is an aerobic organism. It is negative for casein, negative for tyrosine, and negative for xanthine hydrolysis. However, it is partially acid-fast, which is an important distinguishing feature. Nocardia brasiliensis is also aerobic. It differs from Nocardia asteroids because it is positive for casein hydrolysis, positive for tyrosine hydrolysis, but negative for xanthine. Like asteroids, it is partially acid-fast. In summary, Actinomyces is anaerobic and non-acid-fast, while Nocardia species are aerobic and partially acid-fast, with biochemical tests further distinguishing them. Let's review the key reminders about 3 important mycobacteria. Mycobacterium tuberculosis infects about 1/3 of the world's population. It spreads through respiratory droplets and is highly resistant to drying and many disinfectants. This organism is most notorious for causing respiratory disease. On Lowenstein Jensen medium, it produces characteristic cauliflower colonies. Treatment requires long-term therapy, and multi-drug-resistant strains are a major concern. Identification can be confirmed with nucleic acid probes. While in the United States, the skin test is still used as a standard screening tool. Mycobacterium avium complex organisms are found in the environment. They may cause pulmonary disease or disseminated disease, especially in immunocompromised patients. Colonies appear non-pigmented on Lowenstein Jensen medium, and nucleic acid probes provide reliable identification. Mycobacterium leprae is the cause of Hansen's disease, or leprosy. It infects the skin, mucous membranes, and peripheral nerves. The disease is progressive but treatable. Unlike many other mycobacteria, it grows best in armadillo footpads, which is why animal models are sometimes used for study.